Hello everyone, this is Vipul Purohit and I'll be discussing some topics of chemistry of your level. So to begin with, you can see everyone, the name of the topic is there in front of you. Okay, and that is acids, bases and salts. Now if you can have a look at this particular title. Very closely. Okay, I've used some color combinations and there is some reason behind this color combination is. If you can see acids, okay, the coloration is towards reddish. Bases is towards blue. And when we talk about salts, it's a combination of both. Okay, reddish as well as bluish. So I guess you try to understand this now, what exactly it is all about. Okay. So when I talk about salts, it's a combination of reddish as well as blue. So it's very clear that you will be getting an idea that salts are nothing but they are compounds which are going to be obtained by combination of acid as well as base. You getting it? It's so simple as that, right? So we begin with. Now when you talk about the acidic part and the basic part, we have the most important part in this case is the properties. So to begin with, when you start with the acids, they are nothing but they are hydrogen containing compounds. Now when I use the word compound, so that means it's not only hydrogen. Okay, along with hydrogen there will be some other species also bonded with each other. So they are hydrogen containing compounds. Next thing is, now there is going to be a color change. So for that purpose, okay, we need to make use of indicators. Okay, indicators are responsible for color changes. So when we use the indicator that is a litmus paper, indicator as the name says it is going to indicate by means of color change. So the color change is going to be nothing but for a litmus paper is blue litmus getting converted into red. Okay, this is what the acidic character is all about. The property of the acid is all about. Now, coming back to the indicators part. There are various natural indicators. One of the examples that I have is litmus paper. Now, when you talk about litmus, it is actually extracted from a plant that is called as lichen. Okay, it's being extracted from there. The family of this is Thallophyta. And when I talk about plants, so you understand very well that it's going to be what? Natural. The next one, the natural, the most natural, and everybody uses that. Okay, and that is nothing but turmeric powder. Okay, when you talk about a turmeric powder, it's once again, it's going to be a natural one. Alright, so as a result of which, what happens is, you must have seen this, that if there is going to be some stain, okay, which is there on your clothes, okay, obviously, that stain is because of some pulses or which are going to be in along with the spices. So if you take a spicy food, okay, along with that, there will be a turmeric paper as well, okay, the turmeric powder. The second most common, and I guess every one of you will identify with me, and that is the natural indicator is nothing but what? The turmeric powder, which everybody knows it's yellow in color. Now what happens is, if there is a stain on the clothes, now how that stain is, that also I'll tell you, and that is some spicy food, okay, falls on your shirt. The spicy, so that means of course there are some spices, but of course there will be a turmeric powder as well in that. Now if such a stain comes in contact with a soap, okay, of course for washing of clothes we need to use soap, okay, then what happens is it turns yellowish, okay, a reddish yellow, okay, that is what the coloration takes place because soap is basic. So the turmeric powder in basic medium gets converted into what? A reddish yellow color. So a coloration takes place. Okay, once again if you are going to wash it completely with water, Okay, so that the soap gets removed, it once again restores to its original color. Okay, so these are the two most common examples. There are various other examples also, but these are the two most common examples of natural indicators which shows a change in color with the change in the medium. Okay, and that medium can be either acidic medium or a basic medium. Getting me? Right. We go into the next one. The next one we have is like artificial ones or the synthetic ones. In the synthetic case, I can give you the best example is phenolphthalein. Then we got methyl orange. Okay, these are going to be synthetically produced, okay, in the laboratory by means of chemical reactions. So they come under this category and they show a color change. Okay, so I hope you understand the meaning of indicators very well. Okay, they are very important whenever the reaction is going to take place between an acid and a base. And as I told you, that salts are going to be produced only when the acids and the base react with each other, because salts are a combination of acid as well as base. So to indicate the completion of the reaction by means of a color change, that is what the role of indicators are. Okay, one of the best example I gave you was litmus paper, converting blue to red. So this is nothing but acidic. If I make it opposite, that means what I do is I convert in this way a red getting converted into a blue. Alright, so I give you a very simple logic over here, my dear friends, that try to understand this first letter. Okay, that is B. So try to connect it. So it becomes very easy for you to remember. Okay, and that is the blue got B, and that is going to go with what? Base. So base also has the first letter as B. Alright. Similarly, when you talk about red, red is something, it's a signal of danger. And when you compare between an acid and base, who's going to be more dangerous? Oh yes, acids are going to be more dangerous. So that's way you can remember it. So the red signs off for acids. Alright, so this is the best way of identifying the color change or remembering the color change with respect to what? A litmus paper. So litmus paper, when a blue turns into red, it's acidic. Okay, and remember, when a solution is neutral, that means it's neither acidic nor basic, then the litmus has a purple type of a solution color. Okay, in aqueous medium, that means it is in water medium. Otherwise, when you talk about with respect to the acids and the bases, you have got two primarily litmus papers, red as well as blue. So blue to red, acid, red to blue, base, and red remains red, blue remains blue, then the solution is neutral. Am I very clear with all these words? Okay. Right, my dear friends. Now what we do is एक activity करते चलो. Okay, in activity में what we do is कि मैं आपको पहले बीकर में distilled water देता हूँ. Okay, distilled water is a pure water which is neither acidic nor basic. ठीक है? Next बीकर में I give you an acid. Say for example hydrochloric acid, HCl. Okay, उसको hydrochloric acid बोलते हैं. Next में I'll give you a base. Say for example NaOH. Alright. Now I'm giving you a litmus paper. And that litmus paper is only the red litmus paper. I'll write down with red so you understand this better. Red litmus paper. Okay? Now, your activity is using red litmus paper, you need to identify. Okay, ये तो मैंने आपको बोला है कि this contains water and acid and base. Okay? But then, we are not going to say exactly that this only contains water, this only contains acid, this only contains this. No. We are going to identify now which beaker contains what. Alright? So what has to be done is, we have only with us a red litmus paper. So we'll be trying to dip in all the three solutions because we don't know which one is acid and which one is base and which one is water. तो तीनों में हम लोग डिप करेंगे और जिसमें डिप करने से वो रेड लिटमस ब्लू हो गया, ब्लू का बी याद है ना, और दूसरा किसका बी, यस बेस का, दैट इज अ बेस. So it means when I am going to dip red litmus into this, okay, I will be getting what? A blue litmus paper. Okay, it will turn blue. This is an indication that it is a base. You understand this? Okay. Now what you do is, you just wash it with water. Okay, blue litmus paper, you just wash it with ordinary water. Now what happens is, out of the 3 beakers, one is out because I found out, okay, investigation is done, okay, and identification is done that yes, this beaker contains the base, so it's out. Now remains 2 beakers. One contains water, the other contains acid. I got this red litmus being converted into blue, and then only I came to know that and identify that yes, this is a base. So what I do with that blue litmus paper now, okay, and that is I dip into water, I dip into acid. I dip into 2 beakers. Now, whenever I dip that particular blue litmus into a beaker and suppose it turns red, so that beaker is an acid. So it will be this one. Okay, so I'll be dipping this into this. Alright, and then what comes out is once again what? A red litmus paper. So I've identified that this is a acid. So common sense, whatever is left out is what? Distilled water. You getting me everyone? Once again I repeat that. Okay, and how the solution comes is, I'm giving you 3 beakers, no labeling at all. Okay, only I'm telling you that these are the 3 beakers, one of which is containing water, the second one is containing the acid, and the third one is containing the base. And your job is to identify which beaker contains which solution. And I give you 1 red litmus paper. That's it. Do it. So what I do? I take a red litmus paper, dip in all the 3. When I dip it in water, it will remain as it is. When I dip it in acid, it will remain as it is. But as soon as I am going to dip in the base, it will become what? Yes, blue. So I have identified that yes, this beaker is containing a base. So it's out. Now, that red which has converted into a blue litmus now, so that blue litmus, I'll dip it into the 2 remaining beakers. One is containing water and the other is containing the acid. When I dip it in water, that will remain as blue only. But as soon as I dip it into a beaker containing an acid, it gets converted into red. So that means I've identified that that beaker is going to be what? The acid. That's it. The one which remains is distilled water. You getting me everyone? So this is a small exercise that we have done and I hope you like it. And I hope you like it because you have understood the solution. Yes or no? Oh yes. Now my dear friends, we go into the second activity. Now what is the second activity is? And that is, I'll give you a set of acids and base and then we need to find out what are the color changes for different indicators. Alright? So, to begin with, we start with the acids and then we'll go into the bases. Okay? So, to start off with, I have HCl, I got H2SO4, I've got HNO3, I got acetic acid, CH3COH. Alright. So these are all nothing but what? Acids. Hydrochloric acid, sulfuric acid, nitric acid, and it is acetic acid. Or in IUPAC nomenclature, it is called as what? Ethanoic acid. So now, these are the acids. We already know it. But now we are going to identify that with different indicators, what are the color changes that is going to be happening. So we begin with the first one and that is with litmus paper. This I've already explained to you before, but then still as a part of this activity, I'll explain you once again. For a litmus paper, what will happen is the blue litmus gets converted into red litmus. Okay, Blue gets converted to what? Red. This is with litmus paper. Next thing is, we go with phenolphthalein. I made a mention of this, if you remember, when we talk about artificial or synthetic indicators. So, phenolphthalein actually is a colorless liquid. Remember this. Okay. So, there is no change. So, colorless remains colorless. No change at all in the color of the solution. Alright. Next is, we go into methyl orange. As the name suggests, it is going to be orange in color. It gets converted into red. Okay, in contact with these acids. You getting me? So this is what we have. We have a set of acids. We have these indicators, natural and synthetic, and what are the color changes going to take place, right? Now what we do is, we go for the base. But before going towards the base, I'll just give you one important information to you all, and that is this acetic acid. It's actually a weak acid. Okay, it is undergoing what? Dissociation. I come to that dissociation concept as we proceed in this particular chapter. Okay, so bond breaking process, okay, is going to take place slowly. Okay, in case of acetic acid. So it's a weak acid. So generally, the methyl orange is used as a indicator for a weak acid as well as a weak base. Okay, so just try to keep this in mind. And whenever we got strong acids, we generally make use of what? Phenophthalene. Am I clear with this? Okay. Now we go into the bases. The various types of bases that we have. So to begin with, the very first base which needs to come to our mind, okay, and that is nothing but what? Yes, NaOH. Next we have is KOH. Next we have is CaOH twice. Next is MgOH twice. Next we have is NH4OH. Okay, these are all bases. I'll just give you the namings of these compounds. Okay, sodium hydroxide, potassium hydroxide, calcium hydroxide, magnesium hydroxide, and ammonium hydroxide. So once again, we start with the same indicators. So to begin with, litmus paper. Now, when you talk about litmus paper, as I've already explained you before, here what will happen is, the red will be converted into blue. Okay, because this is bases, so the first letter plays a very important role. Next thing is, we go into phenolphthalein. Now, as I told you, phenolphthalein is a colorless liquid. So, I take a colorless liquid, but now, what happens is, as soon as it comes in contact with a basic solution, it turns, come on, can you just imagine what is that color is? Yes, it's the pink one. So, colorless getting converted into pink. This is with phenolphthalein. And the last one is methyl orange. For methyl orange, what will happen is the orange color will be converted into yellow. Orange to yellow. Alright. So this is the indication of what? A basic medium or a basic solution. I'll put a star mark once again over here at ammonium hydroxide with methyl orange. Okay, do you remember the star mark over here? Yes, it's for methyl orange is specifically used for indicating color changes when a reaction involves a weak acid and a weak base. Generally, it is being observed. Alright. So here, the acetic acid is a weak acid. Here, in this case, ammonium hydroxide is a weak base. Okay, once again, the concept is the same, which I'll be explaining you in detail in the later part of this chapter, and that is about dissociation. So when it breaks, the bond breaks, so it is going to go called as dissociation reaction, and it's based on that we say it's a slow dissociation, so base is weak. So we are making use of what? Methyl orange. Otherwise, the strong bases, we generally use phenophthalene. Am I clear with this? So I guess you have understood this second activity also very well.